Okay, welcome everyone. I'm Peter Hoed, and I'm happy to have you all in for our EXIMIOUS Symposium on internal effect markers, a view from immune genetics and epigenetics. And so first, some housekeeping issues. So you have to be aware that this symposium, this event, is being recorded and will be shared on our EXIMIOUS website. And it will be also announced on some social media channel. So if you do not want that your intervention, for the rest, this is fully anonymous, but that your intervention is not shared. If you don't like that, please inform us on info@eximius.eu. And then practical. I try to be your chair for this symposium, but this is an online symposium, and therefore you have not the opportunity to speak, but you have the opportunity to type questions and remarks. And so we have two boxes for that. We have a chat box, and that chat box, please try to keep that for technical issues. So when the sound is not working or the video is breaking and so on. So to inform us on these issues. And on the other hand, we have also a Q&A box, a chat box, where you can pose questions concerning the speaker about the talk for sure. And so try to separate that. And we have two people at the background, Rani and Julia, that help me to follow up these things. And so first, a short introduction on EXIMIUS. Probably you know the EXIMIUS project. It's a Exposome project on which we have different population studies and looking at the human exposome with a specific focus on immune-mediated diseases, and more specific, more even on autoimmune diseases. So it is being funded in a Horizon 2020 program. It runs almost 3 and a bit of years, so since 2020, 1st of January, and we got just a 6 months extension, so it will be finalized at end of June 25. So we have a budget from just not 11 million, and it is a big project. We have 15 different partners from 7 different countries, and EXIMIUS is linked to 8 other EXIMIUS is linked to 8 other exposome projects, all clustered together in the European Human Exposome Network. So our partners in EXIMIUS, that are the 15 partners named here. I will not read them all aloud, but you can see different institutes, also small companies, and spread more or less over whole Europe. And here I show the network of different exposome, European exposome projects. So the European Human Exposome Network, or EHEN, which is built from 9 different exposome projects, of which ours is one, all a bit on different fields in exposome research in Europe. And so you can find more information on this on EHEN on humanexposome.eu. So coming back to business of the day. So today we have our 6th EXIMIUS Symposium. I will not read them aloud, but we had already, therefore, 5 earlier symposia. But if you are interested to review these or if some topics are of interest and you missed it, you can find those recorded on our website, eximious-h2020.eu. So that's our general website. So today we have the 6th symposium on internal effect markers, as I said, with a focus on immune genetics and epigenetics. And so this is the agenda for today. So I'm welcoming you still and introducing the whole symposium. Then we will have a talk by Unni Nygaard on single cell profiling, so the immune focus. Then a talk by Mariona Bustamante, on epigenetic markers, and then talk by Rossella Alfano on linking cholesterol and multi-omic analyses. The first speaker of the day is therefore Unni Nygaard from the Norwegian Institute of Public Health in Norway. She has a PhD in immunotoxicology and molecular biosciences, and she is director of the section in immunology at the NIPH and also responsible for the CyTOF platform. And so I will stop my talk here, my introduction, and I will give the word to Unni.
Yes, thanks a lot, Peter. So I will try to get this up and running. Let's see. So do you see my full screen now?
Yes.
Great. Okay, so thanks a lot for the chance to present our methods and results in this symposium. And I will talk today about how we think that mass cytometry is a promising tool for identifying immune effect biomarkers and thus advancing environmental health and toxicology. But first, a bit about why do we need effect biomarkers in immunotoxicity? So there has been an increased incidence of immune diseases, and as you see on the left graph, that's the prevalence numbers for asthma and allergic diseases, and on the right you see also prevalence increases for a number of different autoimmune diseases. And the reason for this is of course multifactorial, but there has been suggested that environmental factors and occupational factors will have played a part in this, like chemicals, air pollution, food habits, infection, stress, and climate change. And another example of consequences of immunotoxic effects are immunosuppression. The commentary here refers to a paper that reports associations between PCB exposure and antibody responses to the BCG vaccine or tuberculosis vaccine. And as is highlighted, diminished effect of certain vaccines can give really serious implications for part of the world where diseases that are preventable with vaccines remain a major public health threat. So as an overview, the adverse health outcomes from immunotoxic exposure can be increased hypersensitivity or autoimmunity, or it can lead to immunosuppression. And that includes not only reduced vaccination responses, but also impaired protection against infections and a certain number of cancers as well. And the last group of adverse effects are immunostimulation, which is either an exaggerated or prolonged inflammation response and is linked to a number of the inflammatory diseases that we see in our society at the moment. yeah, this was just a comment for this endpoint reduced vaccination responses, because this is really, as we see it, when we see it in humans, we can use it as a surrogate for infection, because it's really the same kind of pathways that are responsible for the response to the vaccine and infections. And besides the actual loss of protection, if you really have a profound reduction in the vaccine response, we see this also as a very good functional effect biomarker or a risk factor for immunosuppression in human studies. So, but unfortunately, in regulatory toxicity testing, there are very few guideline tests that are available to test the different endpoints of immunotoxicity. So in lack of these established guideline tests, in the case of PFASs, EFSA actually in 2021 for the first time used vaccination responses from epidemiological studies to set a limit value for intake. So for these 4 PFASs that you see highlighted, they concluded that the effects on the immune system were considered the most critical for the risk assessment. And therefore we can also suggest that internal effect biomarkers may have an increasing importance also in the risk assessments that we are doing for exposures. And if so, we need to identify sensitive and relative biomarkers of effect that will cover all these different arms of the adverse effects. And that probably means that we need a set of these effect biomarkers to say something about the risk. And to identify new biomarkers that can cover these broad outcomes, we need new methods. And in my opinion, these methods really need to consider 2 important concepts of the immune system. So the first is the complexity of the immune system. This figure is only here to illustrate how complex the immune system is, consisting of a high number of different cell types and a high number of signaling molecules that makes this cell able to talk and communicate with each other, also between different tissues in the body. So therefore, we need methods that capture this complexity or the breadth of the immune responses. And another important aspect of the immune system is that it is in a resting mode until it's challenged. And therefore, we also need methods that can capture the depth of the immune response or how the function actually is affected by exposures. So therefore, we think that high-dimensional single-cell analysis together with explorative data-driven analysis is one of the things that are needed to reveal immune profiles or new cell populations associated with exposures or disease. And this figure is just here to show that the high resolution that is possible with these kinds of analysis, identifying a number of small functional different subpopulations within the major cell types that we have in the immune system. So mass cytometry, or cytof, it's called, is in many ways an extended version of flow cytometry for those who are familiar with that. But instead of tagging antibodies with fluorescent labels, we in cytof we tag them with metals, and this allows for more distinct signals, and therefore we can assess more markers per cell, and that leads to the possibility of looking at the high number of different cell types as well as their function in one single sample. So this is just to show a bit the different stages in the experimental setup. So we start with a complex biological system, such as a blood sample. And then we can stain that with these metal tagged antibodies. And they can bind to proteins both outside and inside the cell. So that way we can stain with markers that recognize different cell types, but also the state of the cell. So cell death, signaling, and different cytokine productions. And when we have stained the cells, we put them into the site of instrument, which is a time-of-flight mass spectrometer, detecting these metal signals. And we typically run 200,000–300,000 cells per sample. And then we really have to rely on advanced computational analysis to figure out the patterns of the data set. And we use unsupervised analysis, data-driven analysis, and machine learning to give us the best picture of what is present in that sample. So the advantages of mass cytometry is that you get this high-dimensional information from a single sample, also in really small volumes, and that you get information on a single cell level and not in a bulk level as you do for a lot of other omics techniques. We do up to about 50 different metal tags antibodies in the same sample, and that allows to detect both phenotypes and functions. And we can do detailed functional characterization of the cells, both in terms of protein expression, but also we can look at signaling, metabolism, and epigenetic markers within the same cells. So for several environmental exposures, there are epidemiological studies suggesting associations between exposure on one hand and immune health outcome on the other side. But these are only associations. So in toxicology, we can use this tool adverse outcome pathways, or AOPs, which is a pathway of key events that can link and strengthen the plausibility of the exposure actually leading to the health outcome. And in our lab, we are now in several projects working to identify cellular mechanisms linking these two by looking for immune cell profiles that are associated either with exposure or with different immune health outcomes or diseases. And these profiles or signatures can also be good effect biomarkers. So today I will show you some results from one of these studies that we have in the lab, looking at PFAS exposures. And I guess most of you know the PFASs, or perfluorinated compounds. They are carbon chains with fluor groups that makes them water and oil repellent and very attractive to use in consumer products, industrial products, and firefighting foams, for instance. And since they are slowly degradable, they accumulate in the environment, get into our food chain, and we are really exposed to these kind of compounds today. So there has been studies that together conclude that several of these PFASs are immunosuppressive. There are animal studies and also human studies showing associations between several of these PFASs and suppressed vaccine responses, and also increased infection frequencies, particularly in children. However, we know much less about the mechanism behind this. So in in this study, we used samples from a cohort established in the EU project Euromix. And this cohort did a really extensive exposure assessment, collecting urine samples, blood samples, and skin wipes, and also had an extensive collection of questionnaire data and food and cosmetic diaries, trying to assess the exposure as good as possible. And in these individuals, we collected viable PBMCs or leukocytes from the blood. And in the plasma samples, we measured a number of levels of perfluorinated compounds. But in this particular study, we chose to look at these 4 compounds that are the most abundant ones in this population. So for the cells, we thawed them, and then we divided the sample in 2. We had one unstimulated sample that was stained with the panel on the upper right with 36 different antibodies, containing both markers for cell types, but also functional and activation surface markers. The other half of the sample was stimulated with p-ionomycin to kick off some cytokine production, and was stained with the panel that you see on the right, down to the right, and where you can see that we also included a lot of intracellular cytokines for the cells. And for the data analysis, they were quality controlled, pre-processed, and living cells were collected, and we also did batch corrections because we run these samples over several batches. And thereafter, we did data-driven clustering by Flowsum algorithm. And in short, that means that this algorithm take the cells that express similar patterns of the markers and cluster them together in these clusters. So on the left side, this is just a visualization tool, UMAP, where you can see that the different major cell populations clustered together, like the B cells, the T cells, the T helper cells, cytotoxic cells, NK cells, and dendritic monocytes. And on the right side, you can see a heat map showing the expression of all the different markers, both for the lineage markers and the functional markers for each of these clusters. So that way we can identify the characteristics of each of the clusters. And thereafter, we used a linear regression model to assess associations between the levels or the concentrations of the 4 different PFASs and the proportion of the clusters, as well as expression of the functional markers in these clusters. So the first set of results for the unstimulated cells, we saw a positive association between the concentration of the PFASs and activated T effector memory cell population. And this population did have markers that pointed towards a Th2 profile of these cells. And at the same time, we saw a negative association between the exposure levels and cytotoxic effector memory population that was associated with a Th1 profile cell. And again, when we looked at the stimulated cells, we saw the same positive association with PFAS concentrations and a T effector memory cell population. And when we here could look also at the cytokine profile of these cells, we saw that they produced cytokines with a Th2 and a Th17 profile. And some of these cells also express FOXP3 and 25, which means that they could be like a regulatory T cell type. So together, this suggests a skewing away from Th1 and towards Th2, Th17 regulatory T cells with increasing levels of PFAS exposure. And we therefore speculate whether this can be one of the explanations that contribute to the observed immunosuppression that we see in epidemiological studies. We also saw several observations between exposure to PFAS and the NK cell or different NK cell subpopulations, but what was common for these NK cell subpopulations were that they were either of a mature or also an activated type of NK cells. So the data suggests that NK cell populations may be stimulated or activated by the PFAS exposures. So to sum up this study, we saw that in an adult population with relatively low PFAS exposure levels, this was a background or a group of adults in Norway that doesn't really have extreme values. But even at these relatively low levels, we saw associations, and we also had quite few participants. So in a way, it suggests that this method is a sensitive way to observe effect biomarkers. And the findings we had points to biologically plausible mechanisms that may contribute to explain the epidemiological reports of the immunosuppression by perfluorinated compounds. And we think that these cell populations also may serve as effect biomarkers in larger studies. I also wanted to show a bit about this other study that we did also in the same cohort, where we investigated associations with phthalate exposures that were measured in the urine samples. This was published in 2021. And we used a slightly different approach in this paper, because here we chose participants that had either a high or a low exposure level measured. And then we compared these two groups. And when we looked at these data, we found that the group that had higher phthalate levels, they had increased innate immune cells, such as activated NK cells and intermediate monocytes, and also increased cytokine production in the NK cell department. The findings that we found with the highly exposed group for the adaptive immune cells were all downregulated, such as we see here for naive T cytotoxic cells and B cells. So I just also wanted to mention what we are doing then in the EXIMIOUS project. So in EXIMIOUS, it is the same kind of concept that is used. We are looking for overlapping immune and omics markers that are associated either with the exposome or disease, and really what is overlapping between these two signatures. But in EXIMIOUS, we are really integrating a number of endpoints to create an immunome. So in addition to whole blood cell profiles done by CyTOF, we are also profiling PBMCs by flow cytometry. We are looking at serum protein profiles, epigenetics, GWAS, telomere length, and secretome or proteomics. And these are endpoints that are done in different labs in Europe. But for the CyTOF analysis that we are doing at the Norwegian Institute of Public Health, we are doing this analysis for a subset of the cohorts that we have in the EXIMIOUS project. So we are doing analysis in a birth cohort from Belgium and Rheinich. We are doing samples from workers occupationally exposed to mineral dust and organic solvents in Romania. And we are doing samples collected for different autoimmune diseases that has been recruited in Belgium. And we are doing, for the site of here, we are doing 400 samples, which is quite a subset of what is done in the whole project as such. So in every site, they collect whole blood samples and they stimulate it on site with a stimulation cocktail that we provided that are designed to stimulate both innate and adaptive immune cells. Thereafter, the cells are fixed and frozen. They are shipped to our institute, and we do sample staining and data acquisition on the site of and data analysis. And this is just to show you again that for these whole blood samples, we are doing a 42 marker antibody panel, which includes both a number of lineage markers, so we can catch all the cells that are present in whole blood, as well as a number of functional markers. And we also use 6 channels for barcoding to have a higher throughput through the instrument. And based on these markers, we can assess innate and adaptive cell populations in whole blood and their function. And we are now really eager to see how those data will look. This is work in progress, and we are soon finished with the whole data set. So we have to come back to the results. And lastly, I just wanted to mention that we, in addition to these cohort studies, have also used the CyTOF in a model that we established, an in vitro indirect whole blood model. And in this model, we use first macrophages as a model of the macrophages in the lung, and they are exposed to, for instance, silica particles. And the macrophages will then secrete a number of factors, such as DAMPs, cytokines, chemokines, and alarmins. And what they secrete into the cell supernatant, we harvest and mix it with a whole blood sample. And after incubation, we look at plasma protein analysis by Luminex, and we look at the single cell changes by CyTOF. And then again, we can do this data-driven analysis, and we are able to identify several populations that are standing out after exposure to this, or this indirect exposure through particles. So what we aim to achieve with this in vitro model is, of course, to look for putative effect biomarkers. And we are really eager to see now whether we actually can see some overlapping biomarkers from the in vitro studies with what we see in the cohort studies, where we have cohorts that have also been exposed to silica particles. And effects in vitro will, of course, also strengthen causality of immune effects by the silica particles. But we also can find relevant, we are looking for relevant in vitro screening models for new chemicals, and we are now testing whether this model can be one way to do that. So to conclude, our studies demonstrate that explorative high-dimensional single-cell analysis is a sensitive tool to reveal immune cell profiles associated with exposures or disease. And we think this is such a powerful tool because we can look at subpopulations of innate and adaptive immune cells in a single sample. So we catch the breadth of the immune response, but also the depth, because we have all these functional markers that are maybe more sensitive and more regular, able to be up and down regulated compared to the major cell populations. And in future, we would like to exploit other sensitive endpoints that we can measure in the cytoplasm, like signaling pathways, metabolism, and epigenetic markers also within the cells. And we can use these results to suggest mechanistic pathways and strengthen causality, but also to identify putative cellular biomarkers for immunotoxicity studies. And we can do that by finding populations that we want to focus more on and make more narrow flow cytometry panels that we can do in larger cohort studies because of the higher throughput. But we can also try to identify cell subsets that we really would like to develop into in vitro exposure models with these solvent cell types, so that we can get some more tests for screening on new compounds. So that was what I had. I would like to thank all the co-workers, of course, in the Unimics and the EXIMIOUS project and the whole EXIMIOUS consortium, which I really enjoy working with. And last but not least, the mass cytometry team at the Institute. And thank you for your attention.
Thank you, Unni, for the nice presentation, very informative presentation. I see that there is one question already in the Q&A from François Huyot, who asks, which antigens activate T cells after chemical exposure?
Can you repeat it?
The whole question, because it seems to be that there is a ... No? No, yeah, it's the whole question. So which antigens activate T cells after chemical exposure?
I'm not sure I quite understand it, but if you ask what's in the cocktail to activate T cells, that's PMA ionomycin in the stimulation cocktail that we have. But I'm not sure if that answers your questions, François.
Yeah, I'm also not sure I understand 100% the question, but maybe he can, François can type a bit more detail in his question. So I had a question about the PFAS experiment, if I may. So I don't see any other question. So you said stimulated and unstimulated cells, but so the stimulation is then in vitro, as you showed in one of the last slides in the EXIMIOUS project, and they were then stimulated with these 4 PFASs as you showed. Is that right?
No, so the PFASs are measured in the individuals. So that's the individual level, and we take the cells from the same individuals, and then we do an unstimulated sample, because that is most correct when you are counting the different cell types, kind of. But if you want to see how they function, you really have to to give some stimulation to see how they behave, and then we can see TH1, TH2, the different subtypes.
And they were then stimulated with a cocktail of cytokines, or were they stimulated with rather or chemokines, or stimulated with the PFASs?
No, so they are stimulated with an immune stimulant that stimulates T cells mostly. So this PMA ionomycin part.
Okay. Then
so the PFASs are only in vivo.
In vivo, yeah. So yeah, that was something I was wondering about. Yeah.
Yeah.
And I do not see any other questions. So people, please ask questions if you have. I got more technical questions. So
Julia.
I think Peter is frozen.
Yeah. So
it seems that I lost you.
Either it's him or me. So I didn't hear you.
I think there was a quick interruption, Peter. We missed your question. If you want to.
So you did not receive my questions. So I was wondering that with CyTOF, and I guess the answer is yes, but it was not 100% clear. So you can, as with FACS, have internal and external factors that you analyze.
Yeah.
Yeah.
Yeah. So we do this permeabilization so that we can also detect intracellular factors. Okay.
Yep. Thank you. And then I have a final look to the chat and the questions. No, I don't see any. No one raised his hand. So, okay. Then I think I may thank you again for the very nice overview of CyTOF, the use of the search for mechanistic, but also really markers of effect rather than of exposure. Thank you. And then I try to share my screen. The next speaker of our symposium is Dr. Mariona Bustamante from Barcelona, IS Global in short, and she runs a PhD in human genetics with a focus on genetic variations, as you can read, and the link to she's a top member of the area in molecular and epidemiology in IS Global. So I will give you the floor, Mariona, because it seems to be that my internet is acting a little bit weird.
Okay, thank you for the presentation. I'm going to share my screen. Okay. A moment, please.
This is feasible, but it's not full screen yet. Yeah. Thank you.
Okay. Okay, so thank you. Okay. So today I'm going to talk about the epigenetic marks of the exposome. So first, I wanted to give a brief introduction to the exposome, but I think that most of you already know it if you are in this European project working on this. So the exposome can be defined as all the environmental exposures that an individual encounters throughout his or her life. So this goes from conception to death. So this is a very broad concept, and we have divided the exposome in three main domains. The external exposome that includes climate, air pollution, or urban exposures. The personal exposome that mostly measures the lifestyle, such as diet, physical activity, or smoking, but also social interactions and chemicals, toxic chemicals. And finally, there's what we call the internal exposome, which are the individual's physiologic and biologic responses to these environmental factors, which are needed to maintain the homeostasis. And this internal exposome has many links to the molecular biology, molecular epidemiology, and toxicology fields. And today I'm going to talk about this, about the biological responses, about the internal exposome. And we can measure this internal exposome using candidate biomarkers or applying omics technologies, which aim us to quantify many different biological molecules, which at the end explain as the structure, the function, or the dynamics of an organism. And these omics technologies can evaluate different molecular layers that you can see them in these figures. So we can evaluate the genome, epigenome, transcriptome, proteome, or metabolome. And all these different molecular layers can be connected to the environment and also to the genome. The connection with the environment has different degrees. For instance, a metabolome can inform us about the internal exposome, but also about the external exposome, the chemical compounds that to which we are exposed. And for instance, the genome, the other molecular layer, has some connections with the environment, because for instance, the environment can produce somatic mutations, and we can use these genomics tools to evaluate them, but the connection is less strong. So in some way, each molecular layer is going to inform us about the piece of this internal exposome. And we can try to combine these different molecular layers in multi-omics analysis to have a broader view of what is happening in response to the exposome. So in this talk, I'm going to focus mainly on the epigenome, but also I will explain some other omics analysis. And the epigenome can be defined as all the modifications to the DNA or to the DNA-associated RNA and proteins that at the end regulate the expression of the genome. So the gene expression, and this finally determines the cell identity and the function of these cells. And as I have said, the genome is in between the genetic and environmental signals. It can capture both signals to produce this biological response. And very importantly, the epigenome is dynamic over time. For instance, this is very clear during fetal development, where there are many massive epigenetic changes in the fetus or in the embryo, even more. And also is tissue specific and cell type specific. And these are things that we have to take into account when we run these epigenome-wide association studies. So there are three main epigenetic marks: the non-coding RNAs, the chromatin, the histone modifications, or the DNA methylation. So the DNA methylation is the addition of a methyl group in a specific positions in the genome, so in cytosines when they are followed by a guanine. And in this talk, I'm going to talk about the DNA methylation mostly, and a little bit about the microRNAs, but basically it's DNA methylation because it's easy to measure in epidemiological settings. So DNA methylation regulates the gene expression, as it is an epigenetic mark, but this regulation is specific to the genomic context. For instance, in the promoter of the genes, when the CpGs are methylated, usually the gene expression is inactivated, but this can be different in other places of the genome. So why do we want to measure the epigenome? So which are the applications? I would say that there are two main applications. The first one would be to understand the biological mechanisms that are connected to the exposome. And the other application is to predict exposures or diseases. Okay. So regarding the first application, what we want to do with this is to understand this mechanism as basic scientific knowledge to see what happens from the exposure to the disease, but also knowing these different steps or gaining insights on this mechanism can give us biological plausibility of the epidemiological associations, and this is important to try to have some influence on public health policies. And here you have a plot of a paper that was published a few years ago in Cell, where that highlighted some of the biological pathways that can be affected by the epigenome, like for instance, oxidative stress and inflammation or endocrine disruption. And one of these pathways was the epigenome, which at the end also can inform about these other pathways, as we will see. So now I'm going to present two studies that we have done in our group, where we have tried to use the epigenetic data and other molecular data to understand the biological mechanism. So in this first study, what we wanted to do is to explore one exposure that was maternal smoking during pregnancy and the placental epigenome. And so what we did was an epigenome-wide association study, and this was done in the context of the PACE consortium. And in the study, we included 1,700 mother-child pairs from different, 7 different cohorts from different places, different countries, mostly in Europe and in the United States. And among these 1,700 mothers, there were more than 300 that were smokers during pregnancy, that they smoked during pregnancy. And placental DNA methylation was evaluated using the Illumina 450K array, which allows us to measure almost 500,000 CpG sites along the genome. Okay. And then also here in this study, we wanted to see if the placental DNA methylation had any connection with birth weight. So here I'm summarizing the main results of that study. So first, we saw that there were more than 400 CpG sites which had different DNA methylation in the placentas of smoker mothers. And this different DNA methylation in the CpG sites was associated with the time, the quantity of smoking. So in the mothers that smoked during the whole pregnancy, the effect, the change in DNA methylation was stronger than in the mothers that just smoked at the beginning of the pregnancy. Okay, so in some of these cohorts, but not in all of them, in a couple of them, we had also gene expression measured in the same placentas. So we could link the methylation of these 400 CpG sites with the expression of nearby genes. And we found that 40 of these CpGs indeed were regulating the gene expression of some genes. And these genes were involved in growth factor signaling, hormone activity, which is crucial for fetal development, also inflammation, that we know that is a pathway that is activated in response to tobacco smoking, and also vascularization, that again, it's another important pathway for the correct function of the placenta. And then we took these 400 CpGs and we checked whether its methylation was linked to the birth weight of the children. And we could find associations in 142 of them. And interestingly, these CpGs that were connected both to birth weight and to tobacco smoking were in regions of the genome that we know that have genetic variation related to birth weight. So in some way, we are seeing regions of the genome, or genes at the end, which both capture the signaling that come from the genome and also from the exposome. And finally, what we did was to compare our findings in the placenta versus previous findings that were published in cord blood to see if the biological response to tobacco smoking was the same in these two different tissues. And we found that the overlap was really very small. There were only 2 CpGs out of the 400 that were statistically significant in the two analyses, in the two tissues. So this tells us that the regulation or the response in the tissues is different. And maybe the pathways that are activated are not so different, but the way to activate these pathways is different in each tissue. And the second study that I wanted to present is a study where we try to identify the molecular signatures of the early life exposome. So this study was done using data and samples from 1,300 mother-child pairs from 6 ongoing birth cohorts from Europe. and from France, Greece, Lithuania, Norway, Spain, and the UK. So in this 1,300 mother-child pairs, we could measure the exposome, both the prenatal exposome or the pregnancy exposome, and also the childhood exposome. And childhood means around the age of 8 years. And for each time period, we measure something as 100 different exposures, which include different families like meteorological variables, air pollution, lifestyle, smoking, also some variables related to social interactions, and different chemical compounds, including the phthalates and the PFAS that have been commented in the previous talk. And then in these 1,300 children at the age of 8 years, we collected blood and urine samples, and we measured different omics. We measured blood DNA methylation, microRNAs, and gene expression using arrays. So we have an overview of what is happening in the whole genome. And also we measure some proteins, serum metabolites, and urine metabolites. And in these 3 cases, we use more targeted methods to measure them. And then what we did was to connect the two data sets, the exposome and the omics. And for doing this, we applied an exposome omics wide association study, or EXIMIOUS, where we tested each exposure versus each molecular feature, one by one, using linear regression models, adjusting for covariates. So we tested something as 30 million associations. Then we applied multiple testing corrections, and we end up finding something as 1,000 exposure omics associations. And then what we did was to try to repeat this analysis by considering different ancestries, by cohort, by considering body mass index. Also, we did some multi-omics exposure networks that I'm going to show them now. And finally, we did the biological interpretation by doing functional enrichment analysis or going to the literature or different public databases to see if what we were seeing had been previously reported or which were the pathways that were affected. So now in this figure, I'm showing you the multi-omics exposome network for the pregnancy exposome. Okay, so here what we did was to take all the associations that we found between pregnancy exposures and molecular features measuring in the children. And with these results, we did this network. We did some clustering to see what we were seeing. And basically, what we see in this network is that most of the associations are with the DNA methylation features. So the DNA methylation layer is capturing the associations with the pregnancy exposome. So in some way, the effects of some of the exposures during pregnancy seem to be persistent in the exposome, in the methylome, but not in the other molecular layers. And I would like to highlight one of these clusters, that is this one in gray, where we found associations between maternal smoking, measured through questionnaire, but also measured through cotinine levels or cadmium levels. That cadmium is one of the components of the tobacco, and also cotinine is one of the components. So we saw in this cluster these 3 exposures, which are related, together with several CpG sites which show different methylation in the children. Okay, maternal smoking, methylation in the children. And in these genes or these CpG sites have already been described in the literature in relation to maternal smoking and also in relation to own smoking. So we were reproducing what it is known in relation to tobacco smoking and methylation. And now I'm going to show you the network that we obtained when we analyzed the results of the postnatal exposome and the child omic markers. And here you can see that we got many more clusters, so many more associations. Indeed, the analysis here is cross-sectional, so the exposure is very close to the molecular signal, and it makes sense that we see more associations. And these associations are not only with one molecular layer, it's not only methylation, but many different molecular layers, okay? And then these associations were clustered with these 7 different clusters, and I would like to just highlight a couple of them. So here in blue, you have a cluster that we call the fish and pollutants cluster that includes several metabolites, which we know that are naturally present in fish, which are the fatty acids that are naturally present in fish, which cluster together with several toxic chemicals that we know that are present also in fish, that the main source of these chemicals is fish, for instance, mercury or the PFAS. So here, the message is that we can use the metabolome, the information about the metabolites, to try to identify the dietary sources of some of the chemicals that we measure in the exposome. And this is the same for this other cluster here in green, where we found that several metabolites that we measure in urine, and which we know that they come from the fruit and the vegetables that we take, were clustered together with some non-persistent chemicals, in particular with pesticides. So again, here we saw that these metabolites were informing us about the presence of pesticides in the fruit and the vegetables. And finally, I would like to highlight this cluster here in yellow that we call weather, where we found that several variables related to weather, like humidity, ultraviolet radiation, or temperature, which are really correlated between them, were clustered with different markers from different molecular layers, including methylation, but also microRNAs, or gene expression, and proteins. And interestingly, one of these proteins was adiponectin, which is a protein that we know that regulates the temperature of the body. So, so again, that here we were seeing more the effect of this exposure on the biomarker. Okay, so as I have explained, we can use the methylation data, or the EWAS, the epigenome-wide association studies, to try to understand the mechanism that link the exposome to disease. But there are many, many different challenges that we have to address during the next years. So the first one is the target tissue. So in epidemiological studies, we only have access to blood and maybe placenta or maybe some other tissue, but not many more. And sometimes we use them as proxy of other tissues. It can be the case, but it cannot be the case depending on the scientific question that we have. Another option would be to use liquid biopsies. Even if we have access to the target tissue, still until now we have been using the bulk tissue. So we have the whole tissue with all the different cell types. And we know that the methylation is cell type specific, and we have to take this into account in some way. Right now, what we are doing is to apply mathematical methods, algorithms, to do cellular deconvolution. So we estimate the blood or the cell type proportions in the tissue using these reference panels and these methods. And then we can use this as a co-adjustment variable, or even we can try to test for interactions to see if the effects are specific to one cell type or another one. But still, I think it would be nice in the future to move to single cell analysis. And also we have used arrays to measure the methylation, which are good because we cover the whole genome, but still they are not sufficient. We are just measuring around 2% of all the CPG sites that are present in the genome. And another limitation in the studies that we are doing is the causal inference. This is a major problem in epidemiology. We try to do it as good as we can by adjusting for confounders. Sometimes we do Mendelian randomizations, but I think it's important to complement our studies with in vitro analysis or with clinical trials if it's possible. Another limitation of the EWAS or DNA methylation analysis is the biological interpretation. So when we detect a CpG site that shows a different DNA methylation in relation to one exposure, we do not know how to interpret it directly. We need to link the CpG to a gene, because for the genes, we know the function. And usually what we do is to link the CpG to the closest gene. But sometimes this is not true. Maybe the CpG is regulating an upstream gene or another gene that we are not considering. So for this, it's really important to have, in some studies at least, data on DNA methylation and gene expression. So in these data sets, we can calculate, we can establish catalogs of expression quantitative trait methylation. So which are correlations between CpGs and genes. And this information can then be used by other studies, by other EWAS to interpret their findings. And here I am showing this catalog that we created in Helix for cis EQTMs in blood for children. And you can find the catalog here together with the catalog of the EXWAS, of the whole analysis where we tested the whole exposome together with the different omics features. So this is a nice resource. And another resource for the biological interpretation of the EWAS is the data coming from the GTEx consortium. So this is an international consortium that I'm sure you know it, that was originally designed to provide gene expression data from different tissues and to understand the genetic regulation of the transcription in these tissues. And now this consortium has also produced DNA methylation data from these different tissues. So they have the expression and the methylation, and these EQTM catalogs are now available for these different tissues. So as I said, the first application of the use of the epigenetic data can be understanding the biological mechanism, but also we can use the DNA methylation or the epigenetic data in general to predict exposures and diseases. So this is not my field, but I think it's interesting to say something about it. So why do we want to predict exposures? I would say the main aim is to provide a better exposure assessment. For instance, measuring the phthalates, which are non-persistent chemicals, it's really difficult. You need many urinary samples to have an accurate measure of the exposure. So if we can define a biomarker based on DNA methylation, or maybe based on other omics that can tell us if somebody has been exposed to these chemicals, it will simplify the analysis in the future. And also we want to use, or we want to predict, or we want to use DNA methylation biomarkers to predict diseases for different reasons. We want to use these information to subdivide, to divide the diseases in different types, to predict the disease risk, but also the disease progression or the response to treatment. So which are the advantages of trying to build biomarkers, predictive biomarkers based on DNA methylation? So one of the advantages is that DNA methylation is tissue and time specific. So it gives some additional information comparing with the genetic predictors, which are static at birth. For instance, we already know our epigenome. So the DNA methylation can bring some information about the preclinical stages of the disease. Also, the working with DNA is much easier than working with RNA, and the DNA methylation is stable over time, and the effects are persistent, as we have seen for the tobacco smoking. And also, there are some dose response between the exposure and the DNA methylation, which makes altogether the DNA methylation biomarkers or predictors good, or a priori interesting to be explored. And to develop these biomarkers, we are not interested in the mechanism. This is not needed. We do not need to understand what is happening at the mechanistic level. We just need to provide a robust association that allows us to classify people based on the methylation profile. So the statistical methods that are behind these are completely different from the US studies that I have mentioned before, where we try to explain the mechanism. And here, finally, what we want to provide is an accurate biomarker, which is sensitive and specific. So in this NICE review that was published a couple of years ago, the authors reviewed the whole literature on DNA biomarkers of exposure or risk factors and diseases. And in this first plot, you can see different exposures or risk factors and how many papers have been published in relation to the use of DNA methylation biomarkers to predict this exposure or risk factor. And you can see that there are quite a big number of publications that use DNA methylation to try to predict age or smoking. Most of these studies are based on DNA methylation patterns in blood, but some others in other tissues. And in this other plot, you can see the papers that have been published trying to describe DNA methylation biomarkers in relation to disease. So on the top, you can see the disease that are investigated, and you can see that cancer is the top disease that has been investigated in relation to DNA methylation. And in the middle panel, you can see which type of cancer was investigated. There are many publications for prostate, breast cancer, and neurological cancers. And in most of the papers, they use tumoral tissue to develop this biomarker. But there are also some papers using other tissues. The papers that, or the studies that use tumor tissue basically aim to predict the progression of the disease, the progression of the tumor. In contrast, the studies that have used other types of samples, I imagine blood mostly, they try to predict the disease, if somebody is going to have the disease or not in the future. And finally, if I have still a couple of minutes, I wanted to talk a little bit about the epigenetic clocks. So the epigenetic clocks are biomarkers based on DNA methylation that aim to predict the age of a person. And the first generation of these epigenetic clocks was published something as 10 years ago. And in this first generation, the aim was just to predict the age and nothing else. And then in 2018, there started to be the second generation of epigenetic clocks that do not only aim to predict the age, but something more related, not the chronological age, but something more related to the biological aging. So they include in this prediction process markers or information such as tobacco smoking, or markers such as inflammation, or lipids profile that can predict you, can tell us which is the health status of a person and the risk of mortality during the next years. So these different clocks, some of these clocks have been trained in blood samples, but others have been trained in many different tissues. It depends a little bit on the clock. And some of them are specific to adults, others are specific to the pediatric population. And the statistical method that is behind these clocks is also different, not depending on the clock. But very briefly, or very summarizing everything, now we can use these clocks to predict the chronological age, and this can be used in forensics, for instance, or we can use these clocks to predict the biological age, either because we use these second generation clocks or because we use the first generation clocks and we calculate the difference between the chronological age and the epigenetic age. And this is called the epigenetic age acceleration, which has been associated with disease risk and mortality. And in our study, in the HELIX study, for which we have these 100 exposures in pregnancy and in childhood, we could calculate the epigenetic age acceleration at the age, around the age of 8 years, so during childhood in the children. Okay, and this calculation was based on the DNA methylation profile that we had measured. And for this, we used some clocks, epigenetic clocks, which were designed in adults. So maybe they are not perfect for the pediatric population, but still they work quite well. And then we associated the exposome versus the epigenetic age acceleration, so the difference between the chronological and the epigenetic age. And we found that maternal smoking during pregnancy, as well as passive smoking in childhood, which is correlated with maternal smoking, and child indoor particulate matter absorbance, so air pollution, in childhood, were associated with an increase in the age acceleration of the children. And in contrast, some pesticides and PCBs were associated with a decrease in the acceleration of the epigenetic age. We think that this positive effect of these compounds on the age acceleration is due to confounding. In the case of the pesticide, we assume that what we are seeing is the positive effect of fruits and vegetables. And in the case of the PCB, we think that we are seeing an effect of the body mass index. And yeah, and also we have here, we develop an air package to calculate these epigenetic clocks in a systematic way from the DNA methylation data. And that's all what I wanted to explain today. Here you can see the different consortia or projects that have given funding for the analysis that I have presented. Here there's the main people that have been involved in these studies. And finally, thank you for your attention.
Thank you very much. It's a very nice presentation and a lot of information. To be honest, it's challenging field. I have a look to the Q&A here, as so from Jagadish, I see here a question. Could you please suggest if there is any method to perform multi-omics analysis using a methylome and metabolome layers? It's a very technical question, I think.
Yeah, so we are now just trying to use multivariate methods to try to integrate the different molecular layers in the same analysis, because what we did, or what I have presented, maybe very fast, was the networks based on the results, not based on the interconnection of the different data, okay? So we are just trying, and we were planning to use maybe canonical correlation or methods such as this one, but we are still trying to see how we do the dimensionality reduction, because for the methylation we have 500,000 CpG sites, and for the metabolome we have 100, 200 features, and trying to integrate the two. We need to simplify this. We need to eliminate some CpGs, and we are thinking about this. And also, we are thinking about how to control for the confounders, because we know that maternal education, the different cohorts that participate in the study have an effect, and we need to eliminate all this noise or all these effects before we try to integrate the multi-omics. So I think I cannot give more details. Also, there are many tools. Diablo is one of the tools that we were thinking about using, or MOFA is another package that could be used.
Yeah. Okay, thank you. And I do not see any new question coming up. I myself have also a more technical question. you said talked about drawbacks or challenges, and you said, yeah, on one hand, maybe we should go because the variation within tissue is more to single cell analysis, and at the other hand, you say, yeah, our arrays are not strong enough. They only cover a very small part of the genome. So if you could choose what you think is the most important one to improve.
I don't know, but I think I would like to go for single cell.
Okay.
Maybe single cell. Yeah, indeed. For instance, in the placenta, I'm really curious to know what happens with the tobacco smoking, the maternal tobacco smoking. If it's a change in the proportion of the different cell types that we see in the methylome, reflected in the methylome, or if it's the methylome in all the cell types that changes. Yeah, and the single cell analysis would allow to see that. I think Peter is frozen. So maybe I can read the question because I see there's another question.
Yeah,
yes, you can go ahead.
Yeah, so regarding the omics analysis, do you use only targeted analysis or also untargeted suspect screening analysis? So for the methylation, methylation gene expression and microRNAs, we did untargeted analysis using arrays. So we are not covering the whole genome, but it was in some way untargeted. For the other omics, the proteins and the metabolome, serum metabolome, we did a targeted approach. We use Luminex and Biocrates. And for urinary metabolites, we use an untargeted metabolomics platform, but we only analyze the metabolites that could be annotated. We didn't go for the non-annotated metabolites because we thought it was too much and we could not interpret that. But now, because in the HELIC children have been followed now at the age in adolescence, so we are doing a visit in adolescence in the same children. So we are following them until adolescence, and we are going to measure the metabolome again. And here we are planning to use deeper metabolomics tools or platforms, and we are increasing the number of features that we are going to analyze.
So we have Rossella Alfano online from the Hasselt University with a background in developmental epidemiology. Today, the presentation will be on unlocking the role of cholesterol in birth weight and the insights from multi-omics analysis. I'm happy to pass on to you. Rossella, thank you very much.
Can you hear me?
Yes, we can hear you.
Okay, and I think you can also see my screen.
Yes, this is great. Thank you.
Okay, thanks a lot for the presentation. It's really an honor to be here today with you. My talk will be about, I will show you the application of multi-omics to a study related to birth weight. And then I will also show you the second part of my presentation, how I went from multi-omics to the exposome. So today, also Mariona showed it very well. We are in an era in which, thanks to advancement in biology, we can measure not only a bunch of molecules inside the human body, but the entire collection of molecules of which we are made up, the omics. So you can see the transcriptome, the proteome, and so on. And also, thanks to technology, we can measure the exposome. So all the exposures that we face during all our life course. And by overlaying all these layers, basically, yes, we can digitalize a human life. as Google Map a bit did, like by overlaying the traffic view with the map view, the street view, etc. We can do the same with the exposome and the omics and have a better idea of health and disease. Mariona showed already like a multi-omics study. What I want to show here, because often we are prone to think about multi-omics are better than conventional biology. I do think they are different. The purpose is different. So in conventional biology, we use a reductionist approach. We look at a few molecules. In single omics, we are hypothesis free and we look to the entire molecular layer. And in cross or transomics, we have an holistic view in which we also try to connect the different molecular layers. However, this comes at a cost. And I think the major cost of multi-omics study is interpretation. It will be very difficult. You saw also in the slide of Mariona you cannot, it's very difficult to grab like one single result as you have in classical biology. Then it's not unrare that after you perform a multi-omics study, you go back to conventional molecular biology, for example, as a validation of your study. And this is what I will show also in my study. Of course, there are many more challenges. For example, like the data are collected from different sources. In the study of Mariona, for example, from urine and blood were mixed. Different techniques, array, assay, mass spectrometry. Also, there is a biological heterogeneity. We are comparing, for example, SNPs and proteins. They have different functions, also different size. Also, there is a high dimensionality that we should take into account. There is large measurement, so million of measurements with small sample size, 100, perhaps thousands. And also the dimensionality differs by omic type, and perhaps you are mixing targeted and untargeted analysis in the same study. Also, you have to deal with missing values, and not only all the statistical approaches deal with it. And also, one of the major challenges is about methods. Before, we had a question about which is the best or like methods to integrate methylation and metabolomics. Actually, there is no answer. And you can see here there is a review, there is no gold standard for multi-omics, and this is really a problem. Broadly, you can differentiate multi-omics approaches in supervised or not, depending if you are integrated also an outcome. But you can see there are many approaches. Mariona mentioned MOFA or Diablo, and you can see here some are displayed, but really it depends on the data and also on the research hypothesis and question that you want to answer. Then I will present the application of multi-omics to a real case scenario, and this is a study that I performed during my PhD, and it's the application of multi-omics to better understand birth weight. But why we focus on birth weight? And here I like to display this picture that show that why we are focusing on the start of the life. Normally, the picture was firstly created for lung function, but it can be applied to any organ. And you can see that there is a line describing the function of the lung, for example, going up, reaching the optimal capacity in adulthood, and then declining with aging. And then if you are exposed to a certain exposure, let's say smoking, during adult life, this decreasing is much steeper. So there is an acceleration, as you can see with the dashed line. However, if you are exposed to a certain exposure, let's say, again, smoking during the in utero life, so if your mother was smoking, your organ will never reach the optimal capacity, so the dotted line. And you can see then that why it's so important to look at the beginning of life. But why I selected birth weight, that also in the study on Mariona was selected as outcome. The reason is that there are many studies also from the first observation of Barker, that is the father of the so-called developmental origin of health and disease, showing that in utero exposures, for example, smoking, are able to act on our body and to determine a variation in the birth weight, for example, by lowering it at birth. And this variation in the birth weight has been associated to diseases later in life, where we are 50 or even older, for example, an increase of cardiovascular diseases. In literature, there is plenty of papers testifying this association. However, what is not known is the underlying biological mechanism. So how this happens. And this is what I was interested in. Now there are multiple ways of performing a study, a multi-omic study, looking at the association with birth weight. However, at that time, I was working with other researchers from Imperial and in a European project called Exposome. And we had available this population of including 4 birth cohorts around Europe for a total sample size of more or less 500 children from Belgium, Spain, Italy, and Greece. And for these children, we had available the cord blood, in which it was measured the entire methylome with the 450K array, the transcriptome with asymmetrics, but it was only measured in a smaller subset of the population, only in Belgium, in the environment cohort, so only for 2,000 children. Targeted proteins measured by Luminex, 16 inflammatory proteins, and the entire metabolome measured via mass spectrometry. Now, how to integrate all this data? One idea came when one of our collaborators in a previous paper in the same population found an association between birth weight and 68 metabolites. Then we thought, okay, we should rely on previous literature. And why don't we use these 69 metabolites as our starting point? At the same time, the PACE consortium also released a paper on a meta-analysis of 24 birth cohorts showing an association of birth weight with 900 CpGs. Then we decided to perform a multi-step study. So in a first step, the so-called, here you see, metabolite-driven approach, we selected in a first step the 69 metabolites previously related to birth weight, and then we correlate them with the other omic layers, so with all the proteins, with all the transcripts, with all the metabolome. And then in a second step, we did the same, but starting from the CpGs. So we took the 900 CpGs related to birth weight, and we correlate them with the transcriptome, the proteins, and the metabolome. Of course, we applied Bonferroni threshold, and then we looked if there were any signals that was in common. On the results, we also did some downstream analysis looking, for example, for pathways. Here I show like main results. Of course, if you read the paper, there are many more. When we looked at the common result between the two approaches, so the methylation and the metabolites driven, we were able to identify over 60 million of correlation, only 7 features that were in common, starting from the metabolome or the methylation side. And you can see them represented in this globe plot. So on the you can see that the 7 features are highlighted in green, and 2 are 2 metabolites, 2 are phosphatidylcholine, basically. One is a metabolite that we were not able to annotate because our methods through mass spectrometry, we were not able to identify any known compound. Then there were 2 CpGs and 2 proteins, periostin and CCL22. While some features, for example, phosphatidylcholine, have been already previously associated with birth weight, others were really new. They were never previously reported associated with birth weight. For example, this is the case of this CCL22. Also, you may have noticed that transcripts are lacking. There is no overlap between the two approaches. However, when we went to pathway analysis, for example, you can see in the picture here represented the pathway analysis of step 1, the metabolite-driven, and step 2, the methylation-driven. There were some pathways that belong to immune system, and one, like the one of chemical signaling pathway, was common to 2 steps. This may mean that perhaps like the post-transcriptional genes that are regulating metabolites or methylation are different, although in terms of pathways, they do the same thing, although via different genes. Then you can see that also we had another idea. 4 of the 7 identified features seems to work kind of together. So the phosphatidylcholine are important also for cholesterol biosynthesis, and the 2 CPGs that we found were located on genes that are involved in cholesterol metabolism. Then we had this idea: why don't we look to cholesterol to see if it is related to birth weight? So what we did, we went to another cohort. Actually, it's EnvironAge. We took samples that were not overlapping with the previous studies for a total of 1,000 newborns, and we measured the cholesterol level, total cholesterol, HDL, and LDL, and we saw if there was any change between small, adequate, or large for gestational age. And you can see the result in the plot, for example, for HDL cholesterol, that indeed there is a difference of babies with being smaller for gestational age, having lower level of HDL cholesterol. And it was the same also for total cholesterol. And this we think is a very nice result because it corroborates the result of the multi-omics study that we performed. Then we thought, okay, cholesterol is important in birth weight. However, we want to know more about which are the exposures that are associated with cholesterol. And then we performed a second study that is actually in something we are working on to test the association of prenatal exposure with maternal and cord blood cholesterol. Of course, in literature, there are many studies about the association of cholesterol with single exposure, although only 2 EXIMIOUS have been previously published and only in adults. And then what catches our attention is that both these 2 EXIMIOUS, you can see some figure from the original paper, they look mainly to diet and lifestyle. So physical activity, for example. While we thought that in EnvironAge cohort, the cohort that we work with in Belgium, we had much more exposure. And we thought to look to the external pregnancy exposome, including information about not only the diet, but also the built environment, the socioeconomical status, high pollution lifestyle, meteorological data, and so on. And also to explore the internal exposome of the mother and the child, so the biological responses in mother and child in relation to cholesterol measurement collected at birth from mother and child. So we went, our study population was based in Belgium, and we had this time almost 2,000 newborns participating in this study. We were able to collect many exposure, a very extensive list, For example, the exposure related to general external exposome, including traffic, noise, green space, air pollution, also specific internal exposure, for example, lifestyle, smoking, diet, health-related, and also internal exposome, including cord blood and maternal blood biomarkers. And here you can see in total we collected 325 variables of the exposome that we grouped according to the main group of the exposome they belong to, for example, air pollution, built environment, diet, and so on. The plot represents the relative variance explained by the single variables, the dots, and by group, the bars. And you can see that, for example, most of the variance in our data set is due to meteorological variables. Also on the top, you can see the number of variables included over the total. So 40% of our data set is made of meteorological variables, but also a pretty good variability is due to health-related, for example, parity, BMI, or clinical data that are collected during the pregnancy, with only 25 exposure that instead are explaining almost 15% of the variance. So pretty good variance. And then you can see on the right, there are two plots. On the top, there is the cord blood cholesterol, and you can see that HDL is a bit higher than LDL. This is absolutely in line with what is reported in literature for cord blood cholesterol, while in adults it's exactly the opposite. LDL is the fraction that is mostly represented, and this is also the case for our pregnant mothers. Then going to the methods, we decided to have two approaches combined together. So on one side, we performed an IXPAS in which each single exposure was associated with cholesterol levels in single linear models adjusted for confounders. And on the other side, we also thought we should go for a more multi-exposure approach. We use Bayesian kernel machine regression using a hierarchical approach. So taking into account also the fact that many exposures are correlated between themselves, and then we group them in correlated group, the one that I showed you before, so air pollution, built environment, and so on. And we only consider an exposure to be identified, to be selected in our final result, only if it is identified by both the models. Using, you can see FDR threshold below 0.05 for the XVAS, and the posterior included probability above 0.5 for hierarchical modeling. And then we use our funding to build network analysis. So here are represented main findings from this analysis. You can see there are several plots for cord blood and maternal blood for the single analysis of CC total cholesterol, HDL cholesterol, and LDL cholesterol. So, on the left, there is the posterior included probability, and you can see if we rank by posterior included probability the different group of exposure that we analyze. At the first position, there is always the internal child for the cord blood and the internal mother exposome for the maternal blood cholesterol levels. And this is pretty, I would say, expected, since cholesterol, it's also like a biomarker that could be included eventually in the internal exposome. What was not expected instead was that the analysis of cord blood cholesterol revealed a pretty high association with dietary variables, while this doesn't happen with the mothers. However, when we go to the single factors that exposure identified, we could be able to identify that this association was driven by eggs. It makes sense because eggs contains a lot of cholesterol, but then also, as in the study of Mariona, we believe that eggs do not mean actually the nutrients that the eggs contain, but the variable that we use is free-range eggs, so non-battery eggs, and they are more contaminated by persistent organic pollutants such as PCB. So we believe that our results indicate perhaps a link with pollutants rather than like the eggs themselves. And also we were quite surprised from also the analysis of maternal blood because we didn't see like lifestyle, for example, physical activity popping up, while some meteorological variables, for example, sunshine, so exposure to sunshine was popping out, and some health-related factors, for example, being a primiparous mother. Then when we took all our results, all our findings, and tried to build a network, what we saw was that there were certain groups. There were 4 clusters, 4 groups. One was the group, the violet one, of the cholesterol in cord blood. Another one was for cholesterol in the mothers. HDL of the mother stand alone, but we were very surprised to see that there was a cluster made of homocysteine and of mother and child that seems to connect the cholesterol of mother and child. So in adult, homocysteine is an independent risk factor for ischemic heart disease, and hypercholesterolemia, it has been hypothesized as an underlying mechanism. In cord blood, homocysteine has been recently associated with reduced carotid distensibility, and therefore our observation of a positive association between homocysteine and total cholesterol in maternal and cord blood may suggest that a similar mechanism may happen also in our children. Then I will go to the conclusion of these 2 studies. Basically, via multiomics, we were able to identify some well-known, let's say, features associated to birth weight, but also new omic features. For example, the CCL22 protein. Also, we were able to generate an hypothesis about mechanisms, the story of cholesterol, and linking cholesterol to the entire exposome. We were able to detect something other than like classical risk factors, meaning a diet or physical activity, BMI, etc., that were related to the cholesterol level in mother and children. Then I like to close my presentation with a future perspective, what we could do more in the future. Actually, it would be nice to link the cholesterol to some at birth with some childhood outcome, like for example, cardiovascular phenotypes or cognition, and to explore if cholesterol is a mediator in this association, and perhaps to also add in this mediation effect, like the also some omics of the one that we found in our multi-omics study. And these are the list of the most closer collaborators and also the institution that participated through this study. Thank you.
Thank you very much, Rossella. Yeah, I'm sorry, but I was, yeah, my computer was blocked, so I was totally out of the air, and luckily Julia did take over. So I will have a look here in the chat, because I also didn't get the whole story, therefore, but I see one question here. Thank you for, oh no, that's a message from Julia. But Yeah, maybe I missed it, but there seems to be a clear link between the cholesterol of the mother and of the child. But when you say cholesterol of the child, is that then cord blood or really child blood's cholesterol?
So for now, we only looked at cord blood. So what we saw is that there is, of course, a correlation, but between the mother and the child, the cord blood cholesterol, although this correlation is not that strong.
Mm-hmm.
And this also makes sense because during pregnancy, the cholesterol passes. It's actively transported. It doesn't pass through the placenta. So it makes sense that during pregnancy, especially in first or second trimester, there is a stronger correlation, while as the fetus develops and also its liver starts to work, like the correlation decreases. And also we see it in our study. I think maximum correlation that we see is 0.15.
Yeah. No.
So there is, but it's not that strong.
Not that strong, no.
And also, like you can see it also, the exposure of the mother and the child are pretty different. There are like some, for example, ferritin, it pops out in both the mother and the child, but the rest are really different. Meaning that perhaps the mechanisms are also different happening.
Okay, thank you. Yeah. And I have a look here in the Q&A. I do not see any upcoming questions. So thank you very much for the very interesting presentation. And yeah, the somewhat, for me, surprising is maybe too big word, but at least very interesting results with this cholesterol study. Thank you. And then I try to share my screen again. So we just had the final talk for the day from Rossella, and in an attempt to wrap up, so we had an exercise on cytoph in identifying functional and cytological markers of exposure and disease, and then with Mariona we had really an in-depth view on epigenetics, mainly in young children, but also the clock, I cannot say gene, but the clock markers, and several technical ways in trying to link omics and disease and exposures, markers of disease and exposure. And then here, yeah, the link between cholesterol, birth weight, and the link also with the external environment was explained. So thank you very much. Very interesting for me, at least a very interesting seminar on different issues from the internal Exposome. So thank you, Unni, Mariona, and Rossella. I thank also Julia to take over when my computer was blocked, and Rani to help with follow-up of technical issues. Certainly, thank you, audience. I didn't see you, but at least I've seen that quite some people were online, so that's always nice. And certainly, I have to thank our local people setting the seminars up together with the working group in EXIMIUS, but Katrijn and Manosic are driving forces in this, and all the other EXIMIUS partners. So thank you very much, and hope to see you in our next seminar. But also please have a look in the chat where Julia has posted a small comment still on the feedback. So please, if possible, fill out the feedback. It's very useful for us to look into what are your interests and also what we can do better in our next seminars. For instance, getting a computer that works and doesn't bother. So thank you very much and see you later. And
Thank you.
also
Bye-bye.
thank you all the speakers. Bye-bye.